last bit of information that we're going to cover in Unit 3 are the organisms that are in the family Enterobacteriaceae. And this is not an exhaustive list by any means, but it's still a lot of information, so just take your time and go slow through these slides. Okay, the first one is E. coli, which I'm sure everyone has heard of. Diseases that it can cause, UTI, and if you remember from a previous lecture, E. coli is the most common cause of UTI and kidney infection in humans and can lead to secondary sepsis. It can also cause meningitis. It's one of the most common causes of meningitis among neonates, where it would be acquired in the birth canal before or during delivery. It can be recovered from wounds, bacteremia, again, which just means presence of bacteria in the blood. So in adults, this is primarily going to be from a genitourinary tract infection or a GI source. So in other words, it's going to be secondary to some other type of infection. And then gastrointestinal or diarrheal disease, and we talked a little bit about enterohemorrhagic E. coli or O157H7. So within that group of the pathogenic E. coli, there are a few more that we're going to talk about. So enteropathogenic E. coli, seen primarily in infants and children. So you'll have outbreaks in hospital nurseries, daycare centers. Cases in adults are not seen that often. In a case of enteropathogenic E. coli, the stool sample itself will have mucus, but not blood. And this can cause chronic diarrhea, more common in developing nations. Okay, then we have enterotoxigenic E. coli, which is sometimes referred to as traveler's diarrhea, where the patient will have profuse watery stool, again without blood. It's transmitted by contaminated food and water. It occurs in adults and especially in children, and especially in tropical and subtropical climates, and especially in developing countries. Enteroaggregative E. coli, the actual pathogenic mechanism for this one is not known, but the patient will have watery diarrhea that can become prolonged. The mode of transmission for this one is not very well understood. Okay, enteroinvasive E. coli produces dysentery, which is an infectious disease marked by necrosis, inflammation, and ulceration of the lower part of the bowels. So there's bowel penetration with this one, invasion and destruction of the intestinal mucosa. Patient will have watery diarrhea with blood. These infections can occur in adults and children, but they're not very common in the United States. They're identified by DNA probes. Most of these strains are non-motile and do not ferment lactose. Then we have enterohemorrhagic E. coli. So this is the one that we've already touched on just a little bit. So this is serotype O157H7, associated with hemorrhagic diarrhea and hemolytic uremic syndrome, where the patient will have a low platelet count, hemolytic anemia, could lead to kidney failure. This one is potentially fatal, especially in young children, and it's associated with undercooked hamburger, pasteurized milk, and apple cider. It'll start with a watery diarrhea and progress to bloody diarrhea, but you will not find white cells in the stool associated with this one. This one can be fatal, especially in young children and elderly adults in nursing homes. Then we've got diffusely adherent E. coli. It's a cause of diarrhea, but it can also cause UTI. There's some virulence factors associated with E. coli. Invasiveness, so it has the ability to get into the host tissues. It produces cytolysins, also known as hemolysins, which will kill immune cells. It'll inhibit phagocytosis. Fimbriae and pili, so this allows for adherence to the epithelial cells, which means that it would not be washed away with urine flow. And then we talked previously about this Shigella-like toxin, so it's similar to the toxin produced by Shigella, which is why they call it the Shigella-like toxin. So it produces a cytotoxin that's Similar to the Shigella toxin, it's called virotoxin 1 when we're referring to 0157H7, but it also produces another toxin called virotoxin 2 that's slightly different. These toxins, the mechanism by which they work is just to damage cells. And then ESBL, which stands for extended spectrum beta-lactamase. So this is an enzyme produced by some strains of E. coli that make them resistant to certain classes of drugs. And there are other organisms within the family of the Enterobacteriaceae that can produce this, so this is not specific to Enterococcus. I'm sorry, not Enterococcus E. coli. Okay, lab ID, coliform, which just is kind of a general term meaning that it's a lactose fermenting intestinal commensal. Colony morphology on your EMB, it has that very specific and characteristic green metallic sheen, if you remember from pictures that we've looked at previously. On the McConkey, it usually appears as a pink colony, which if you remember from the differential qualities of McConkey, that means that it's a lactose fermenter. And then sorbitol McConkey, which we've mentioned previously. Also, your 0157H7 will be colorless, and all of your other strains of E. coli will be pink. So that would be done, you would use that sorbitol McConkey in stool cultures specifically. Its MVIC reaction is positive, positive, negative, negative. And if you remember from the previous lecture, MVIC is it's a combination of a few tests. So the I stands for indole, M is methyl red, V is the Vogts-Proskauer, and C is citrate. So that means it's positive for indole, positive for motility, negative for Vogts-Proskauer, and negative for citrate. Its TSI reaction is, most of the time it's going to be A over AG, which means it produces acid in both the butt and the slant because it utilizes glucose and lactose, and then the G is means that it produces gas, if you remember that. Or it can rarely be seen as K over AG. Its urease and PAD are both negative. The MUG test, which we talked about also in the previous lecture, you can use it for presumptive identification of E. coli. So your EHEC or your 0157H7 is negative, but most of the other species are going to be positive. And the positive results in that fluorescence under UV light. And then it is also motile. And there's a picture of the organism on the EMB that it's showing that characteristic green metallic sheen. Just a few images to represent the transmission of the organism. Okay, next is Klebsiella, so some of its general characteristics, usually found in the intestinal tract of humans and animals or free living in the soil, water, and on plants. They say it has a distinctive yeasty odor. There's 4 major species, so Klebsiella pneumoniae is the most commonly isolated in the clinical lab. It possesses a polysaccharide capsule which protects the organism against phagocytosis, but it makes the colonies very moist and mucoid, so its colony morphology or how it looks on the plate, it can be very like wet and runny. And this also plays a role in antibiotic resistance. This one is non-motile, so if you remember from the previous lecture, one of the general characteristics of the Enterobacteriaceae is that they are motile, but Klebsiella was one of the exceptions to that. All species are resistant to the antibiotic ampicillin, and some are now showing increased resistance to other classes of antibiotics. Some of the diseases that it is associated with, so it's largely associated with opportunistic and hospital-acquired infections, so lobar pneumonia, especially in the hospital setting. So, and lobar just means of or relating to a lobe, so it would be related to the lungs. It's a cause of UTI. It can cause wound infections, bacteremia, and liver abscesses. Some of its virulence factors, so it produces something called KPC or Klebsiella pneumoniae carbapenemase. So this is another enzyme that causes resistance to the class of drugs known as the carbapenems. And this enzyme was first identified in Klebsiella pneumoniae, but it's now being seen in other enterics also. So the name has changed to carbapenem resistant Enterobacteriaceae, or CRE. And then this one is also capable of producing ESBL, which is another marker for antibiotic resistance. Lab identification, it's a coliform, just meaning that it's a lactose fermenting intestinal commensal. The colony morphology, again, can be very mucoid or wet or runny looking. It's pink on your EMB and McConkey, meaning that it is a lactose fermenter. Its TSI is A/Ag, and one of the claims to fame for this organism is that it produces lots of gas. Rarely you can also see a TSI reaction of K/Ag. Its MVIC reaction is negative, positive, positive, so it's the exact opposite of E. coli, and it is PAD negative also. It's non-motile, generally speaking. Some species or some strains can have variable motility though. Lactose positive, most are slow urease positive, and then it's ornithine negative. And here's a picture of some of those, and this is not even one of the worst ones I've seen by far, but some of those like mucoid, runny looking colonies, and this is on the McConkey auger. Okay, the next organism is Enterobacter. So it's comprised of 12 species, with Enterobacter cloaceae and aerogenes being the most common. It's reported as one of the genera listed in the top 10 most commonly health care acquired infections, usually associated with contaminated medical equipment devices such as respirators. It can also be ingested from water, vegetables, and food products. It can be isolated from wounds, urine, blood, CSF. Some of its major characteristics, the colonies can resemble Klebsiella on McConkey. It's motile. Most of its biochemical reactions are going to be similar to Klebsiella, except this one is ornithine positive. It is ONPG positive, which, if you remember from the previous lecture, means that it is a delayed lactose fermenter. Its MVIC reaction is the same as Klebsiella, TSI, A/Ag, and the PAD is negative. And here's a picture of Enterobacter aerogenes on the McConkey plate. You can see those dark pink colonies, meaning that it's a lactose fermenter. Okay, Pantowea and Hafnia. So Pantowea used to be thought of primarily as a plant pathogen, but it's now being associated sporadically with traumatic injuries from objects contaminated with soil, and also some healthcare associated infections have occurred from exposure to contaminated IV fluids. This one can produce a yellow pigment on the media, so it's colony morphology. And then Hafnia, there's only one species, Hafnia alvei, has been linked to gastroenteritis. It's occasionally isolated from stool cultures, but usually in conjunction with other organisms. It resides in the GI tract of humans and other animals. So infections are associated with consumption of contaminated food, meat, and dairy. Most infections are identified in patients with severe underlying disease. It has a delayed positive citrate reaction, which is one of its major identifying characteristics. Okay, Serratia. There are 8 species, but Serratia mercescens is the main clinically significant species. It's associated with opportunistic infections. It's an opportunistic pathogen, a lot of times associated with nosocomial infections, especially UTIs and infections in the respiratory tract. It's implicated in bacteremia outbreaks in nurseries, cardiac surgery, and burn units. It can be recovered from wounds. Its lab identification, its TSI is mostly A/A, but can sometimes be K/A. Some strains can produce gas. It's ONPG positive, so it's a delayed lactose fermenter, and PAD negative. Some species are chromogenic, which means some of them can produce a pigment. Pigment is called prodigiosin, and in strains that produce that pigment, the colony morphology on the plate will be very red colonies. It's resistant to a wide range of antibiotics and disinfectants. It's capable of survival under very harsh environmental conditions. And this one, it's just because it's so resistant, inherently resistant to so many different antibiotics, when we perform susceptibilities on this organism, we only test it against a few different antibiotics. I don't know what kind of media this is, but that is an example of Serratia that produces prodigiosin, so it's given that very bright red color. Okay, the next one is Proteus. This one is normal intestinal flora, so it is considered an opportunistic pathogen. It can cause UTIs and wound infections. Proteus mirabilis and Proteus vulgaris are the major species that you would see recovered in the clinical lab. It is motile, and this one is a little bit different with its motility. It swarms on non-selective media such as blood agar plates. So what that means is instead of like it will take over the entire plate. I've got a picture I'm going to show you in just a second, but it kind of grows out from the center, and it will end up swarming or taking over that whole plate, so that if there are any other organisms on that culture besides the Proteus, the likelihood of recovering it is not great, just because everything on the plate will be contaminated with Proteus. It has a distinct, what they call a burnt chocolate smell. It's a non-lactose fermenter. It's methyl red positive. Its Vaux-Proskauer reaction can be variable, as can its citrate reaction. It is rapid urease positive, PAD positive. It produces hydrogen sulfide. Your Proteus mirabilis has a negative indole, a positive ornithine, and the TSI is K over A plus G. MVIC is negative, positive, variable, and variable, whereas your Proteus vulgaris species is indole positive, ornithine negative, so those two reactions are opposite from Proteus mirabilis. TSI is A over A plus G, and its MVIC is positive, positive, negative, positive. And here's that picture of Proteus swarming the plate. So what they have done is subcultured a colony of Proteus from another plate. So they have taken the loop and stabbed it right into the center of the plate there. You can see the little stab mark. And then, so this plate has only been inoculated in one specific place, but the organism swarms. It kind of looks like the rings on a tree. It ends up taking over that whole plate. So if there were any other colonies growing on this plate out in this swarm, we would not be able to recover them. Proteus is kind of a aggravation sometimes. Okay, Morganella. Morganella morganii is the only species. It's found throughout the environment. It's considered normal flora of the GI tract. There are documented cases of UTI and neonatal sepsis. It's commonly recovered from stool specimens in patients with diarrhea, but they haven't really determined the clinical significance of that. This one is also a non-lactose fermenter. It's urease positive, PAD positive. TSI is K over AG, and its MVIC is the same as E. coli, positive, positive, negative, negative. Okay, and there's what he looks like on the blood agar plate. And just kind of as a little mini quiz here, can you determine what kind of hemolysis that is? It's beta. You can see that kind of thin zone of beta hemolysis around the colony there. Okay, the next one is Providencia. This one also is a non-lactose fermenter. Its TSI can be K over A or K over AG. It just depends on the species. Its MVIC is positive, positive, negative, positive, and that's the most common reaction for most of the species, but there can be some variance in the citrate reaction in a couple of species. Urease reaction can be variable, and then the PAD is positive. Providencia rectigeri is a pathogen of the urinary tract. It can cause nosocomial outbreaks. It's also been known to cause diarrheal outbreaks in travelers. And this one is urease positive. Providencia stewartii can cause nosocomial outbreaks in burn units. It's been isolated from urine cultures as well. And this is another one of those that just kind of has inherent resistance to antibiotics. Okay, there's a picture of Providencia stewartii, and that is on a McConkey auger. And you can see, we said it was a non-lactose fermenter, and look at the colonies on the McConkey plate. They have not changed the color of the media. They've not changed to that dark pink color that would be indicative of a lactose fermenter. They've kind of remained the same color as the original media. So that's how you know it's a non-lactose fermenter. Okay, the next one is Citrobacter. It's an inhabitant of the GI tract associated most commonly with nosocomial UTIs, but can also be recovered from wounds, the respiratory tract associated with bacteremia, endocarditis, and meningitis. There's at least 12 species. The most common that you will see in the clinical lab is Citrobacter caseri, freundii, and brachii. And each one of those species will have different embic reactions, which are listed there on the PowerPoint. Lab ID, TSI, K/A+G, or A/A+G, and that's just because you can see that variation just because it's such a slow lactose fermenter. Some are K/AG or A/AG, so some species do not have the hydrogen sulfide production. It can be confused with Salmonella if it's isolated from stool cultures, so that would be very important to differentiate. It is lactose positive, lysine negative, arginine positive, ornithine variable. The urea can be variable also. And that's just some of the tests that you can use to differentiate it from Salmonella. And then it's also PAD negative. Here's what our Citrobacter looks like on McConkey. Okay, next, Salmonella, which we've already spoken a little bit about, but this one is always considered a pathogen. So a lot of these organisms in the family Enterobacteriaceae are considered normal flora in the GI tract. This is one that would always be considered a pathogen. So it causes typhoid fever and other enteric fevers. So that consists of a prolonged fever, dissemination to multiple organs. It occurs more commonly in tropical and subtropical countries, results from ingestion of contaminated foods, and the onset of symptoms is usually 9 to 14 days after ingestion of the contaminated food. It can also cause acute gastroenteritis, which would be more self-limiting in healthy individuals. This one can occur, or you'll start to see onset of symptoms about 8 to 36 hours after ingestion of the contaminated food. Most cases have been linked to poultry, milk, eggs products, and handling of animals. So that's usually, more commonly, it's cold-blooded animals like snakes, turtles, etc. And I think, if I'm correct, it is still illegal to own turtles as a pet. Or to buy them from a pet store. Pet stores can't sell them, and it's because of that, for the risk of Salmonella. But Salmonella has a very high infective dose. It can also cause bacteremia. There's a carrier state following Salmonella infection, so patients who recover from the infection can still carry the organisms in their gallbladder and can shed them in their feces. So because of that, it can be infectious to others still, even though the patient is, or the host is not exhibiting symptoms, they can still pass it. Okay, some virulence factors associated with Salmonella produces an enterotoxin, which causes gastroenteritis. It is able to transverse the intestinal mucosa, and they don't really know how that happens. It has fimbriae, which helps with attachment. So antigenic structures. This is called the Kaufman-White scheme. So it's a classification based on surface antigens. We can serotype the different strains based on these antigens that they have on the surface of the cell. So the O antigen, and we've talked about this before, the O antigen is the cell wall antigen that is heat stable. The H antigen is the flagellar antigen, which is heat labile. VI capsular antigen, which is heat labile. So this is the capsular antigen. So it's like the K antigen that we have spoken about previously, but it's specific for Salmonella. So VI is technically a subtype of the K antigen. Few strains may possess the VI antigen, but it helps to prevent phagocytosis. So this one is important in identifying Salmonella typhi. Okay, your lab ID, TSI is K over A plus G. The PAD is negative, urease is negative, MVIC is negative, positive, negative variable. So again, that variable citrate reaction can depend on the serotype. It's lactose negative, but it's lysine, arginine, and ornithine positive. And if you remember from previous lectures, the media that is selective and differential for Salmonella and Shigella, we can use the SS media. The SS media, XLD, bismuth sulfite, and hectawin. It produces clear, colorless, so non-lactose fermenting colonies that have that black center if the media has an indicator for hydrogen sulfide production. And then we can also use that Kaufman-White scheme or antigen serotyping, so it classifies the serotypes based on those surface antigens. And then the Widal test tests for antibodies against the organism to determine if the patient has or has had enteric or undulant fever infection. But you also have to take into account if the patient has had a typhoid vaccination, because they would have produced antibodies in response to a vaccination. Okay, transmission through contaminated food, milk, and water. Contact with a carrier. So this is what happened with the case of Typhoid Mary. So it was stored in the gallbladder. In carriers, it gets excreted in the feces. Removal of the gallbladder is sometimes the only way to treat those patients who are carriers. It's a natural hosts for this organism can be cold-blooded animals, rodents, birds, reptiles, and that's different species, but not Salmonella typhi. It's also associated with improper disposal of sewage, poor sanitation, lack of modern water treatment systems. Okay, and then some species, Salmonella enterica, which is a human pathogen, and Salmonella bongori, which is an animal pathogen. So enterica is divided into 6 subspecies. Then it gets further divided into serotypes, and that would be based on that antigen testing, with each one of those serotypes having unique virulence properties. Salmonella typhi is the primary identifiable serotype that's associated with typhoid fever. And there's a picture of those black hydrogen sulfide producing colonies, and this is SS media. Just a picture of Salmonella with his little fimbriae, and a couple pictures just to represent some transmission. Okay, our next one is Shigella, which we've also mentioned previously. This one is another one that's always considered a pathogen, transmitted human to human. So humans are the only known reservoir, not disseminated in nature, and this is through contaminated food and water. Flies can carry it, so oral fecal transmission is usually how that contaminated food and water is occurring. This one has a very small inoculum to cause disease, so fewer than 100 organisms are needed for infection in healthy individuals. So this one is considered highly communicable, highly infectious. So even in healthy people, people who have very robust immune systems, if they come in contact with even just a small amount of a small inoculum of Shigella, they're going to be sick. Okay, this one causes shigellosis. So this shigellosis begins as watery diarrhea. Patient will have fever and abdominal cramps, and this is a progressive disease that can lead to dysentery, where the epithelial cells of the bowel will be invaded. So it's an infectious disease marked by inflammation and ulceration of the bowels with diarrhea that contains blood and mucus. And then bacteremia can also occur, but that one is not common. Lab identification. So the specimen that we would recover this from would be stool. So this one is not part of the normal GI flora, though. So again, it would always be considered a pathogen if it's recovered. It is susceptible to the acid pH of stool, so it should be plated as soon as possible to help recover this organism. It's a non-lactose fermenter, which means that it will appear as clear colonies on the differential and selective media that we use to recover it. The TSI can be K over A or K over AG, and then your MVIC variable for indole, and then positive, negative, negative. It is non-modal, which means that it would lack the H antigen. And for serodiagnosis, so it's important to serotype if we recover an isolate of Shigella. So there's 4 species, several different serotypes within each group. All possess the O antigen, which is that cell wall antigen. Some have the K or capsular antigen. And remember, that can interfere with the detection of the O antigen. It's O and PG variable, urease negative, lysine negative, hydrogen sulfide negative, PAD negative. And it's been found to be very similar to E. coli through DNA hybridization techniques, but it's kept separate just for medical reasons that are related to categorizing diseases. There's a picture of Shigella on the Hectoin plate, and if you remember, Hectoin is used for identification or recovery of Salmonella and or Shigella. The media starts out as this green color, and Shigella and Salmonella both are non-lactose fermenters, which means that it will not utilize the lactose in the media, so it will not change the color of the media, so the colonies remain green. And Hectoin also has the hydrogen sulfide indicator in it, but Shigella does not produce hydrogen sulfide, so you will not see those black colonies. Okay, the next pathogen in this group is Yersinia. There are 3 species considered human pathogens. So the first one is Yersinia pestis, which is the causative agent of the plague or Black Death. This is a disease primarily of rodents transmitted to humans, usually through the bite of an infected rat flea, but it can also occur by handling infected animals, inhaling infectious droplets, and through ingestion of undercooked meat. In humans, plague can occur in 3 forms. So you can have bubonic plague, which most people have probably heard of, where the patient will have a high fever. It forms places under the skin, especially near the lymph nodes and groin. You can also have septicemic plague, which is when the bacteria spreads to the bloodstream. And then you can have pneumonic plague. So this would occur secondary to bubonic. With this one, there's going to be lung involvement, but it can be a primary infection if the organisms are inhaled. This one is considered a Class A bioterror agent. Okay, Yersinia enterocolitica. This is the most commonly isolated species in the clinical lab. It can be found in a wide variety of animals, so pigs, cats, and dogs are just a few of those. Infection can be acquired from contact with household pets, but it's primarily through ingestion of contaminated food or water. It causes diarrheal disease, which can mimic symptoms of appendicitis. So it has the ability to invade the intestinal mucosa. It produces enterotoxins. Again, it's through ingestion of contaminated food or water or market meat. And this one has the ability to survive refrigeration. So it can withstand cooler temperatures. And actually, if we're trying to recover this one in the lab, it has a special media that we use for it. And we incubate that plate at room temperature. Okay, Yersinia pseudotuberculosis. found in a wide variety of domesticated animals, such as rodents, birds, and rabbits. Human infections with this species is rare, but when they're recovered, it's usually associated with close contact with infected animals or ingestion of contaminated drink or food. The disease with this one is usually self-limiting in humans, but it can be fatal in animals. So as long as the host has a pretty healthy immune system, they might have mild symptoms and they can recover fairly quickly. A lab identification for Yersinia, it's gram-negative short, plump rods with what they refer to as bipolar staining. So it gives it a safety pin appearance on the gram stain. It's non-modal. Urease positive except for Yersinia pestis, lactose negative, ONPG positive, mannitol positive, catalase negative. CIN is the media I was talking about that we use for isolation. We talked about it in a previous lecture, but it produced, so this is the media that we would leave sitting out at room temperature because it likes those slightly cooler temperatures. And we're looking for those bullseye colonies on the CIN as indicative of Yersinia. It's capable of growing in a wide temperature range, so it can actually grow anywhere between 4 and 43 degrees Celsius, which means that refrigeration would be ineffective as a means of prevention. And then Yersinia pestis, your TSI reaction is K/A, and MVIC is negative, positive, negative, negative. And then this is an image representing that bipolar staining or safety pin appearance. You can kind of see it there. So that is the end of the lectures for Unit 3. We covered a lot of information, so just make sure you reach out if you have any questions.